This presentation is over the drug Lovastatin and how it works to lower cholesterol. So first, what is Lovastatin? Lovastatin is an HMG-CoA reductase inhibitor, which is the classification for the statin drugs. Lovastatin is used together with diet, weight loss, and exercise to reduce the risk of heart attack, stroke, and chance of heart surgery for those that also have heart disease or for those that have a high chance of developing heart disease. Lovastatin may also be used to decrease the amount of cholesterol or other fatty substances within the blood. And Lovastatin is classified as a natural type 1 statin, natural meaning that it's not synthesized within a lab, and type 1 means that it contains a naphthalene ring. So for the cholesterol biosynthesis, first of all, it starts off with the CoA thioester group of HMG-CoA and it reduces to an alcohol in NADPH dependent catalyzed by HMG-CoA reductase producing mevalonate. Next, mevalonate gets converted to phosphomevalonate with the help of the enzyme mevalonate 5-phosphotransferase. And then the phosphomevalonate gets converted to 5-pyrophosphomevalonate with the help of the enzyme phosphomevalonate kinase. Next, the molecule undergoes an ATP-dependent carboxylation reaction. So the 5-pyrophosphate mevalonate gets converted to isopentyl pyrophosphate with the help of the enzyme pyrophosphomevalonate decarboxylase. Next, the isopentyl pyrophosphate gets converted to dimethyl allyl pyrophosphate with the help of the enzyme isopentyl pyrophosphate isomerase. Next up, there is a series of condensation reactions. So the first one, prenyl transferase catalyzes the condensation of dimethyl pyrophosphate and isopentyl pyrophosphate to produce geranyl pyrophosphate. Then prenyl transferase catalyzes a second condensation of geranyl pyrophosphate and isopentyl pyrophosphate to produce farnesyl pyrophosphate. Then the squalene synthase catalyzes the third condensation of 2 farnesyl pyrophosphate molecules to form squalene. Next up, we have squalene epoxidase that catalyzes the oxidation of squalene to produce 2,3-oxidosqualene, and here we start to see the ring structures begin to form. Finally, we have oxidosqualene cyclase that converts the 2,3-oxidosqualene to lanosterol, which is the first steroid produced, and here you can see the 4 ring structures that are the base of what will eventually become cholesterol. So the lanosterol then converts to cholesterol through a 19-step process, and that is how cholesterol is synthesized. So Lovastatin inhibits high cholesterol by blocking the HMG-CoA reductase enzyme that is needed to make cholesterol. Statins have a higher binding affinity to the HMGR, and it binds to the L domain of the HMGR to sterically impede HMG-CoA. So it won't begin the process of making cholesterol. Thus, the structure of the Lovastatin does get changed in order to have it become active.